Here is a clear, structured, and detailed explanation of the different types of animal disease models commonly used in biomedical and pharmaceutical research. This is widely used in toxicology, pharmacology, oncology, immunology, neuroscience, and drug discovery. Animal disease models are essential tools that help researchers understand disease mechanisms, evaluate drug efficacy, test toxicity, and develop new therapies. These models mimic human diseases through various approaches. 1. Natural, spontaneous disease. Models. These are diseases that naturally occur in animals without experimental manipulation. Examples: dogs developing naturally occurring osteoarthritis, cats with natural diabetes mellitus, rodents developing mammary tumors, non-human primates with spontaneous hypertension. Advantages: closely resemble human disease progression, provide realistic clinical settings. Limitations: hard to control variables, limited reproducibility. 2. Induced, experimental disease models. These diseases are created intentionally to mimic human pathology. This is the most commonly used type. Types of induced models. A. Chemical-induced models. Chemicals or drugs are used to induce disease. Examples: STZ, streptozotocin. Type 1 diabetes, DMBA, MNU. Cancer models, CCL4, liver fibrosis, LPS, sepsis, inflammation models. B. Surgical models. A surgical procedure induces disease conditions. Examples: MI model, myocardial infarction, by coronary artery ligation in mice, nephrectomy, chronic kidney disease, spinal cord injury, neurological models. C. Diet-induced models disease produced by specialized diet challenges. Examples: high-fat diet, obesity, metabolic syndrome, high sucrose diet, type 2 diabetes, high cholesterol diet, atherosclerosis. D. Infection-induced models pathogens are used to mimic infectious diseases. Examples: influenza virus, flu models, Mycobacterium tuberculosis, TB models, SARS-CoV-2 infection, COVID-19 models. 3. Genetic, transgenic disease models. These animals have specific genes modified to mimic human genetic diseases. Techniques: knockout, gene deletion, knock-in, gene insertion, CRISPR, Cas9, gene editing. Transgenic animals overexpressing human genes. Examples: No-D mouse, autoimmune diabetes, APOE knockout mouse, atherosclerosis, P53 knockout mouse, cancer, APP, PS1 mice, Alzheimer's disease, SCID, nude mice, immunodeficiency. Advantages: highly reproducible, ideal for studying molecular mechanisms. Limitations: high cost, genetic background influences results. 4. Xenograft and patient-derived models. Human tissues or tumors are implanted in immunodeficient animals. A. Xenograft models human cancer cells injected into immune-deficient mice. Types: subcutaneous xenografts, orthotopic xenografts in organ of origin. B. PDX, patient-derived xenograft models. Fresh tumor tissue from patients implanted directly into mice. Advantages: maintains human tumor characteristics, used in precision medicine. Limitations: requires immunodeficient animals, time-consuming. Orthologous, simulated physiological models. The disease is similar in animals and humans due to similar physiology. Examples: pigs for cardiovascular disease, dogs for osteoarthritis, non-human primates for neurological disorders. These resemble human pathology naturally due to similar anatomy and physiology. E. Environmental models disease caused by environmental exposures. Examples: exposure to pollutants, asthma models, cigarette smoke, COPD, UV exposure, skin cancer, noise exposure, hearing loss models. 7. Behavioral, Neurological models used to study psychiatry and neurodegenerative disorders. Examples: forced swimming test, depression, MPTP induced, Parkinson's disease, canic acid, epilepsy, social defeat model, anxiety, PTSD. 8. Combination, hybrid, models. Combination of genetic, environmental, surgical, and dietary factors for complex diseases. Examples: genetic plus high-fat diet, obesity plus metabolic syndrome, chemical plus surgical, liver fibrosis plus bile duct ligation, genetically modified plus infectious agent, autoimmune models.